So, can I start now? I guess. So thank you everyone for coming to listen to this great seminar today. I'm very honored to host that seminar as a student chapter in UIUC. So today's speaker is Alejandro Keller. Alejandro Keller is a research scientist for the University of Applied Science Northwestern Switzerland. He has been working in the development of aerosol technology since 1997. One of his main interests is the quantification of particulate matter emission using a metric that takes the dynamics of atmosphere into account. This has led him to several developments, including the microsmoke chamber, the fast summer carbon totalizer, and the oxidation flow reactor. And today's the title of the speech will be the organic cooling unit, an all-in-one system for reproducible generation of secondary organic matter aerosol. Yeah, please welcome him.
Yeah. Thank you for the introduction and thank you for the invitation. I'm very honored to be here, and I want to talk to you about this device that we built here at the university. And I mean, Judith mentioned I have been working with aerosols since '97, and at that time at the university there was no lecture for aerosols, so I was working at the solid state physics department, and everything you needed to have a whole lab just to measure anything. So I think that's something that marked several people from our group, not only me. And we try to make these small practical devices that you can use even if you have limited space, or you can just take them with you to the field. Anyway, I'm guessing the invitation came because we published this paper already 1 year ago, and yeah, I mean, I'm happy I chose AST to publish it because I can give the lecture now, and I have seen that it somehow had a nice impact. A lot of people have been downloading it, and probably that's also how it catches your attention to invite me to talk about this. But I did suggest to have the name of the paper, the name of the presentation, but since we have some time, I would like to talk to how we arrived to this idea. So an alternative title for this presentation could be like why we ended up building the organic coating unit when what we really want to do is measure the impact of wood burning emissions, and this is something that still interests me a lot, and that we are also working on this. But one thing led to another. I mean, spoiler alert, it's all about the funding that you can get. So you need to change a little bit the direction of your research depending on where the funding comes. So at that time, it was like about the mid 2000s, and we were in Switzerland interested in the impact of wood burning in the country. How important was this? And I have here some examples. Maybe not all of you think about the different possibilities for wood burning. And in this first example, this is a festivity in Zurich, where I live. And they do this once a year, and you can say they are burning the winter away. So they have this puppet here, and they do this big thing, this huge fire, and then you can see all the smoke going out. I mean, there are several festivities like this in Switzerland. Swiss people they like to burn things a lot. So what you're seeing here is coming out. You probably have way more air than what you need for the combustion, and it usually tends to make the particles so they are mostly organic compounds. It doesn't mean there's no soot in there. It doesn't mean that there's no other thing in there, but the biggest part of that will be organic compounds. Exactly the opposite, and this is a picture I found on the internet from a traditional Finnish sauna. I like a lot when the colleagues of Finland, they talk about wood burning, because they always have a sauna to test, and this is a very bad one. And probably here you have the opposite situation where you don't have enough air, and the result is that you are creating a lot of soot, and this is why everything looks so black. And of course, it doesn't have to be any of these situations. It's both are very bad. You can come to an optimum, and there are very modern installations like this one, where they will, let's say, close enough to burn perfectly the wood. I mean, still you have emission from this because biomass and all these materials, they not only have carbon, they have also minerals and ashes. And unless you have a filter system, you will be emitting them even if your combustion is perfect. So what's happening at that time is that based on the installations that were in Switzerland and based on the quality of air that we're measuring, there was something that was a mismatch. So from the measurements that were done on site, you could not predict what was actually happening. And this is a picture I took from Polscher Institute, PSI Switzerland. They do a lot of atmospherical chemistry studies. And this is in a valley in Roveredo, where it's heated a lot with wood burning. So what you're looking at here is none of the situations that I was showing to you earlier. So this is also aerosol particles coming from wood burning, but they are not primary emissions. If you work in aerosols today, it's almost impossible not to know about secondary organic aerosols. And here I have a very simplified view of what's happening. You have your combustion, and when you are burning your material, you are producing some gas phase compounds, but also some particulate matter. And this all goes into the atmosphere, which is a huge reactor. And then you have the solar light, but also some other reactive species that help you do some atmospheric processes. And this will change this gaseous carbon into particles. It will lower the volatility so they can condense in other particles or even nucleides and produce more particulate matter than what you have at the beginning. And what was happening is, if we go to the site and measure the organic carbon against the elemental carbon ratio of the emissions, the primary emissions, you would see that it's always much more elemental carbon that soot than organic carbon. Whereas in the atmosphere in Switzerland, there is much more organic carbon coming from wood burning emissions that soot. And this is, you see, it's an incredible change between what you measure here and here. Not everything is to blame on these atmospheric processes. It's also the way these particles are sampled, as usually they do it taking a heated filter. And this, of course, changes the partition as well. So some organic material that will be primary, it's not measured at the emission site. Anyway, we were trying to figure out the best way of measuring these particles. So what you have probably seen, and I apologize because I only put example of European environmental chambers. You have these chambers which are basically just big Teflon bags, and you can have some mixture of gases inside and maybe some particles. And then either you expose them to solar radiation, like this one's in Valencia or this one in Jülich, or you have it in an enclosed space where you can also change the temperature, and then you produce your own solar radiation using UV lamps. And I mean, this has gone so far that there are also this so-called portable chambers. I mean, but still, I mean, this is several meters. I mean, there are many cubic meters of air here, maybe 10 cubic meters of air. So there's still a big chamber. And what you want to study is what I told you before, how this organic carbon undergoes chemical reaction to produce more particles. These experiments take a while. I mean, already the preparation, imagine the preparation for an experiment in one of these chambers may take you 1 day just to prepare the chamber for the experiment. You have one of these, then maybe you can do one experiment per day. And in this one, if you start very early and then you finish very late, maybe you can do 2 experiments per day. And what you do is you sample, let's say you sample a whole cycle of wood burning, you put it there, and then you see what happened. But then you don't really know what was happening during the whole process. So for this reason, at that time, back there, during this project, we tried to do an oxidation flow reactor. At that time, they were not so popular like right now. We wanted to do this oxidation using UV light as fast as possible. And you can see that the residence time in the reactor is about 10 seconds. So in the atmosphere, you will have these processes that will take you hours, days, maybe 1 week. If you have a smoke chamber, like the ones I showed you before, maybe you can accelerate these processes to something like 5 hours. Most oxidation flow reactors that's out there, they have a residence time for about 1 or 2 minutes. And this one is 10 seconds. So this is a big difference. And at that time, there was nothing like this. There's still nothing like this out there. I mean, most of the oxidation flow reactors are designed for ambient concentrations. And this one is designed to be used with just slightly diluted emissions. So to say, in the stack of your wood burning, you have something like 10% CO2. And then you dilute a factor 10, you have like 1% CO2. And ambient would be 500 ppm of CO2. I mean, but not everything comes from the wood burning. So these are very high concentrations. So we use milligrams per cubic meters of particles instead of micrograms per cubic meters that you will have in the atmosphere. And we use ppm of the precursor substances instead of ppb. And then let's say if we want to do ozonolysis, we may go as much as 80 ppm of ozone instead of some ppb of ozone in the reactor. So this is a brute force attack, and this is what we thought was suitable to measure these wood burning emissions. We did continue with this work, and I'm trying to give some historical context. At that time, the European Union was trying to consolidate standards because there were many different standards to measure wood burning emissions. And this is just not perfect. I mean, and many countries didn't even do a type approval test of the installations. So we started a project which was called ENPME test. And the goal of the project was to establish a common European standard to measure wood burning emissions. Not the best standard, just something everybody could agree on. And Europe is very diverse, so it's very difficult to come to the same. I remember the discussions during this project. I mean, we sometimes some group will change the people that were working, and we had to start the discussions all over again. So basically, what we did is at the end, what was suggested is something very simple. You take a filter, you heat it to 180 degrees, you sample the emissions there, but you also measure the gaseous species that go through the filter, the organic carbon, and then you do gravimetric. That means that you weigh the filter before and after. There are some restrictions, and this is why we had to do it like this. And one restriction is that this has to be easy and cheap, because you cannot ask for a test that will cost you tens of thousands of dollars for a single stop to a manufacturer that sell products for, let's say, $1,000, $2,000. So this is something you need to adapt the technique so that it's cheap enough that it's still viable for this application. If not, then it will not go through with the standard. And this was a success. I mean, it took 14 years from the beginning of the discussions to now, but since last year, finally, this type approval testing was approved. We were in this project, but we were not discussing this kind of work. We were trying to I mean, this is the goal, to have a common European test method, and it has to be relevant for air quality, but also the cost and simplicity should be appropriate. But what we were discussing is that everybody could agree upon, that is something that everybody in Europe would agree upon, is what this standard is not good enough. We need to go for something better. And what we were pushing for is to also take a look at atmospheric aging already at the type approval testing, try to simulate this atmospheric aging. So you can see here, we have, after the project ended, we published this little paper with what we proposed could be a good method, which means you oxidize with this small reactor your emissions, and then you take some kind of metric to measure the particulate mass. We propose to measure total carbon, but here the example that I'm showing you here is measured with the mass of the secondary organic aerosol. We have a filter. We only let the gas phase go through the filter, and then we oxidize them. And I'm showing you this just so that you can see that this reactor actually let you see transient processes. I mean, this is half an hour more or less of what the stove is burning every time. You have here a cold start, and the cold start at the beginning produces a lot of organics. Then you have here a stable phase where you produce less, you have a better combustion, so you produce less, and at the end, the combustion is also slightly worse, so you produce more organics. So you can see these typical cycles where you have a lot at the beginning and maybe more at the end, and here at the end was not so pronounced. So this is what you can see, and I put these lines because these lines are from a study done in another stove by another group using one of these big smoke chambers, and you can see that we are in about the same order of magnitude. I mean, it was not the same stove, but at least we can agree that in the amount of secondary organic aerosol that can be produced by one of these. This is a modern stove, not a bad stove. Anyway, our goal, like I said, was to make this compatible with test bench and field measurements, not necessarily to make it the most scientific relevant approach, but something which is relevant from the air quality and the scientific point of view, but it's also compatible with a very pragmatic approach. At that time, this did not go through because it was considered that this oxidation flow reactors were still very young, not available enough. The situation has changed. I'm showing you here, for instance, 2 oxidation flow reactors that you can buy. I mean, one from Aerodyne, the PAM chamber, and one from the CATI, the CATI oxidation flow reactor. And I mean, these are already portable enough, and you can measure it. Like I said, these are not like ours. These are done for ambient concentration, not for high concentrations. We wanted to have high concentrations because then our system would be also compatible with this gravimetric metric that was being used at that time and is still being used today. So you can have a more modern approach, but you can also have both things, like the traditional gravimetric approach or a more modern instrument to measure your emissions. Well, what happened is that this project ended, and so the funding that we had at that moment for studying wood burning also ended. But we were already a little bit in the standardization track. So we team up with metrology, with the Federal Office of Metrology from Switzerland, METAS, and we tried to generate just what we knew and the tools that we had available to generate some standard aerosols. So in the field, what you have is you have some emissions, in this case, emissions from cars, which are emitting soot, and then you have the atmospheric processes, and then you're having your secondary organic aerosol particles, which probably are covering some primary particles like soot. So in the lab, what we propose is, instead of having a car, you take a generator. In this case, this is a diffusion flame generator where you are producing soot. You try to produce the blackest soot that you can, almost 100% elemental carbon. You have some conditioners here. And then you add, you add here a little bit of a precursor substance, which you control here. With this device, you control the concentration that you're adding. Then you go through this oxidation flow reactor. And then from there, you can go to all your instruments to measure. So we took a reactor and added to this setup. At the end, because of all this that you got, looks like this. So I mean, you cannot see it, but the reactor is somewhere around here. And for that, this is the active charcoal denuder that is mentioned here. So this is, I mean, it's still, compared to what you have in a laboratory, what you have a smoke chambers, is still a very let's say, practical setup, but you can see that it's getting complicated. Here are some results. I mean, you have here your original soot is this one. This is your size distribution of soot. And as you start adding alpha pinene, and this is the ratio of the mass of alpha pinene to the mass of black carbon of the soot. As you start to add this, then you start to increase the particles in size because you are coating them, and you start to make your distribution narrow and narrow because you are condensating on this, and this is what happened. And I want to show you this because even though we have such high concentrations of precursor substance and this very strong UV light, we are managed to not create a nucleation mode back here, which could be a problem. We want to have our secondary organic aerosol coat that soot. So we were managing to do this. Here you can see it also. Yeah, here the geometrical mean diameter of your particle, and you can see that, and I will talk about this later, it goes a little bit down, and then as you coat the particles, it starts to go up in size. I mean, from 90 nanometers to 200 nanometers, that's a lot of coating that you're putting in. And here you can see this geometrical standard deviation. So it's going down, which means that your size distribution is getting narrower. And here you can see the coating here, elemental carbon to total carbon. You start with over 80% elemental carbon, and then you will end up having less than 10% elemental carbon. This is just the carbon which comes from soot to the total carbon. If you want to look at the mass, this is what it is. This is the mass of the organic coating divided by the mass of the soot. So you are putting 15 times more mass into each particle. Like I said, this is a lot of coating that you're putting into the particles. But once you have this application, once you have this standard, and let me go back, I mean, you can see here the error bars. I mean, these points were reproduced day after day after day. And then you can see the variability is actually quite well. I mean, it's very difficult to do the experiment several days in a row and reproduce the points with these precisions that they managed to do in metas here in Switzerland. So once you have this system, then you can think about several applications. For instance, here we did a study with the University of Bern and metas, of course. I mean, this whole installation is in metas, and where they have this in vitro deposition studies. So they have here a vial, and they put some cells inside it. I mean, here. And then this is actually a system that we also built at our laboratory. Here is the reference. This is the deposition chamber. So you have all the cell cultures in here. And what's happening is you have some electric field that helps you deposit the particles in your cells very homogeneously. So this is done to do this health studies in vitro, and this is called a air liquid interface study. And here are some results from that study. I mean, this was financed by the Federal Office of the Environment here in Switzerland. And you have here this LDH release, and this is LDH is a marker for cell damage, for cytotoxicity. Here you have your incubator control. You have always a particle free air control. I mean, this was not exposed to particles or to anything. This was just left where it was in the incubator. This was exposed to air without particles, to the gas phase of the emission, but not to the particles. And then you start here with soot particles, which are 30 nanometers or 90 nanometers. And then you coat it with secondary organic aerosol coming from alpha pinene, which is a biogenic secondary organic aerosol, what you will producing. And here some anthropogenic kind of secondary organic aerosol. And we were trying to see if there was a difference in cell damage or cell response into this. I mean, usually I would say, especially if you look against the control with particle-free air, I mean, usually we can have more indication of cell damage when you have the anthropogenic precursor than that you have the biogenic precursor. This was a pilot study. We wanted to show that it was possible. And this pilot study, but this pilot study, we thought it was kind of complicated. So we wanted to make this a little bit more simple. So we thought that we could revisit our setup because like I said, this is at Metas and this is working very stable and you can go there and do your experiments, but sometimes you need to do experiments somewhere else. And if you take the set of somewhere else, or so if somebody wants to replicate it, they cannot just take your the set points that you use, they have to characterize everything from scratch. So this kind of takes away a little bit this kind of experiment as a standard for generation of particles. If you have at the same time, same place all the time, okay, but if you want to transport it, it will be difficult. So we thought that we could take some of these components and make them in kind of a standardized device that we could reproduce and that could just travel. So we can put this in a box, send it somewhere. And the idea is that you will be able to reproduce the exact same set points that were designed by Metas in another location. And this was done, like I said, this pilot study that we did in Switzerland led us to propose this in some metrology, European metrology project, which was called Aerotox, which finished some years ago, last year, I think. And this was a European collaboration with several other groups from England, from Germany, from Finland, from Sweden. I'm sorry if I don't remember all the partners at this moment. Anyway, the device looks simple. You have here some mass flow controllers that will help you dose some organic compounds. You can have 2 organic compounds with those. You have here your primary particles, your seed particles, or simply just air. You can humidify them or just leave the humidity as it is. And then you go through 2 mixing stages. So you can mix 1 or 2 organic compounds. The first organic compound, you set the target of the concentration that you want, and the device will control it automatically. Here, no, here for flexibility reasons, because we want also this to be able to dose, let's say, some gas mixture that you have. We decided not to go with the control loop. I mean, usually we just do one BOC, but it's possible to do 2, or it's possible to mix here something different. Here you can see the concentration of the BOC that you're putting. You go to the oxidation flow reactor, and then you go to your experiment. So it looks quite simple. It's very compact. Here, what you see here, this is the oxidation flow reactor. So it's very small. It's even smaller than the one that we had previously. You can see the length of the reactor. I mean, this is 18 centimeters. So the reactor is maybe, I don't know what is, maybe 12, 13 centimeters, where you are exposed to UV light. And we did several of these instruments. So here are some results from this system. I mean, this compared to what we had. So you can see here, I put the concentration throughout several days, and you can see that day after day, you can reproduce the points. You start with uncoated soot, and then as you increase the ratio of alpha-pinene to soot, then you start to see the coating. So the ratio of the elemental carbon of the soot to the total carbon, it's been reduced all the time. And like I said, you start with something like 90% elemental carbon, and then you go here all the way to something like 10% of elemental carbon. What you want to go for most applications is here, but you can even go beyond that and create very, very big particles, which are mostly organic carbon. Here you see also this size of the particles, how with the coating the size is increasing as you would expect. And what's even more better is that you can also control other properties of the particles. For instance, if you want to have a particle which is designed for some specific optical properties, and here I choose a single scattering albedo, for instance, 0 would mean that your particle is 100% absorbing. 1 would mean that your particle is only scattering light, it's not absorbing light. So as you coat your particles, you go from practically 100% absorbing to practically 100% scattering because of the increase of organic coating that you have there. So you can actually create particles which have some specific chemical and physical properties with this, and this can be done also in a reproducible manner. And if you see here, we always plot the ratio of alpha pinene to elemental black carbon, the equivalent black carbon, because you can do this in 2 different ways. You can have different concentrations of elemental carbon and then adjust your amount of alpha pinene. So you can go to the same set points. Let's say these 2 are similar with a different amount of alpha pinene. You need just to have the right equilibrium in this ratio. So if you want to have very large particles and coat very fast, then you can have a diluted soot. But if you want to have a more controlled coating of your particle, then you can have, for instance, undiluted soot from this device. And then here, you take the first dilution step away, and then you can have a much more controlled coating of your particles. And here you can see, this is the same curve that you can see here. So you start from uncoated to coated particles. Instead of going in the picture that I showed you before, you will go to a single scattering albedo, which is 0.8. And here we arrive only to a little bit less than 0.2. So we are just at the beginning of this coating process. So we are just dosing a little bit to coat the particles slowly. So we start with the, like I said, the naked soot here. It's mobility diameter is 92 nanometers. And when we do the first coating, we see that the particles actually reduce in size instead of increasing due to the coating and reducing size. And you can see here also that these particles collapse a little bit. If you coat a little bit more, then they become even smaller and even more compact. And if you coat here a little bit more, they change from these fractal aggregates to a compact particle, and actually very fast. And this is a nice result because this kind of images you may find in other publications, and there is the debate if the restructuring of the particle happens when the coating is taking place, or if it happens when you put the particles into the microscope, because you put then these particles in vacuum, the organics evaporate, and this may cause a restructuring of the particles. So it was not clear from when this restructuring came. In this particular case, because we have some online instruments, we can see that the size of the particles is being reduced. So this collapsing of the particles is happening during the coating process and not when you are looking at your particles in the microscope. So this is some kind of interesting results that we were not expecting, that we can achieve because we have this very controlled system. We work with the University of Basel, and they did some chromatography studies, because it's not obvious that when you increase the amount of precursor substances into the reactor that you will produce the same chemistry. So they did this chromatography analysis and also principal component analysis, and they arrived to the conclusion that the secondary organic aerosol, even though we were increasing the quantity of precursor substances, is always same. And this is important because if you want to do these health-related studies, then you want to be sure that you're studying always the same substances. So if you coat more your particles, or you're coating it with a different substance, then you don't know if the toxicity change happen because you are using different chemicals, or if it happens because you increase the amount of coating in your particles. So this is also a nice result. What you can also do is skip at all the soot particles, so you don't have any seed particles. You dose your compounds and then you do nucleation particles. So here you have an example of nucleation particles from alpha-pinene SOA. And then you can see the mobility diameter is very small, 10 to 15 nanometers. And we are doing this in dry conditions. And because of the way our reactor is built, the chemical path that we are using is mainly ozonolysis. But if you change this and you humidify your sample, here you see we are increasing the relative humidity, then you open up different reaction paths. So you are producing secondary organic aerosol. And here you see well, I don't see it here. Here's 50 ppm of alpha-pinene. So we are actually here. And instead of having around 10 nanometers of particles, we can have particles which are pure secondary organic aerosol, and they're about 100 nanometers in size. So this is also something special about this reactor, that you can produce very small nucleation particles, but you can produce 100% secondary organic aerosol particles, which are quite large, and this may be of interest for certain applications. What has happened since we published the paper is that we had some feedback from our partners. We built several instruments. We built 4 instruments of this first generation, and we decided that we could improve it. So we have a little bit of change. You see these 2 modules here. This is where you would have your precursor dosing, and we changed it so that you can also control the temperature of your precursor substance. And this opens use possibilities to use, for instance, very volatile molecules that will be difficult to dose in small quantities, or you could have also organic compounds which are maybe solid at room temperature and would be difficult to dose. So we decided to go for this range. I mean, probably we could go to higher temperatures as well with this setup. We have limited per software. And this 10 degrees is because below 10 degrees centigrade, then you need a much more complicated setup in order to cool your particles down. But I mean, this is already gives more flexibility to the device. Well, I mean, these are. Here are some applications, and the first application, and if you notice, I talked several times about the optical properties, and this is not just like that. It's because our partners at Metas, they were looking at absorption photometers, and they wanted to have a standard aerosol to test them, to challenge them. So they use our system to generate this standard aerosol and to compare different devices. And if you want to look at the data, here's the publication. You can see it, but there are some filter-based instruments and also some instruments that measure in situ. So I think these 2 are filter-based. This is a photoacoustic system, and these 2 are interferometers, which should be the ones that have the less artifacts of them all. So you can see that the absorption that should be measured the same, and they're all seeing the same particles, and they are reporting especially the devices that have filters. They have some artifacts. They are calibrated with a certain particles, and they report something different. So this asks for and this will be our next application. This is our next application. This asks for a more thorough calibration procedure. So right now, There is a European metrology project starting this year, and will go for 3 years. We are 10 partners in this project, and the aim of this project is to develop a new European standard for calibrating this, let's say, elemental carbon measuring systems. This is one of the reasons why probably this is not so well established as a standard metric in all measurement stations on the cities and roadsides is because you have these differences depending on which device you use. So calibration is very important. So you can see here already in this infographic from the project, that's the base of the project, is that you will have some reference soot aerosols and you will have also some coded soot, and try to figure out what's the best way of interpret the information that you have for your devices in order to be able to track not only the absorption coefficient of the sample, but also a certain elemental carbon mass out of that. So this has not yet started. This will start this year. And one of the instruments that will be a key for this project is our coating unit. Also, and this is very nice, I asked for two of the groups that are using these prototypes. I mean, like I said, we built in total 7 units, and most of them are owned by different groups right now, and they're using it without our help. I mean, this also says that these units are well built and easy to use, because we don't get a lot of calls of what they need to do in order to make them work. So they're working fine. So I had to ask, what are you working on? So for instance, this is a group at the University of Basel, and they told me they are using this generation of secondary organic aerosol also for toxicity studies. This is also, I am guessing, a liquid interface similar to what I showed you before, but they also have this technical note. I actually didn't know they published it, and they are trying to figure out how, if you use filters to have a sample and then you transport the sample to your laboratory or somewhere else, how does your sample is affected? And you can only do this if you have a very well-defined source that that lets you compare what happens if you have a fresh sample or sample that you produced 10 days ago, for instance. So they produce this technical note, and actually I only heard about this last Friday, but I think that's very exciting. Also, and these are the better slides. I got this from the University of Lund, and they are right now, I think they just finished a study, and they want to see the effect of soot particles on birds that lives on the city. And what they were explaining to me is that birds have actually a very efficient respiratory system, and being very efficient, very direct, not with all this little channels that we have in our respiratory system, is that they probably also much more efficiently take up soot into their lungs. So which means that they may be more affected than us. So the question that they're asking, and what they're doing here is an in vivo study, is if birds are more sensitive to soot in terms of toxicity that we are. And they also show me these slides. They're looking at oxidative stress and oxidative stress, but also an inflammation, and also the overall condition. So this is the study that they're doing. I know that the measurement campaign just finished. They did send me some other slides also to show how the generation of secondary aerosol is. I mean, they did a lot of mass spectrography on the samples, and from what they told me is that they're very happy with the instrument, because it actually produces secondary aerosols also with a chemistry which is what you would expect from what is found in the atmosphere. Anyway, like I said, you can have a generator to produce some seed particles. They don't need to be soot, they can be something different. And then you can dose some biocids into it, and then you can produce so coated particles with this. You can also leave the seed particles away and just dose your precursors and produce some homogeneous nucleation particles that can be actually quite large. But what you can also do is you can have a source of aerosol and then don't add any precursor and use this as an oxidation flow reactor because it has an oxidation flow reactor in, and then you will see what this secondary organic aerosol production of the source that you're studying is. So you can also use it, of course, as a oxidation flow reactor, but it is much more than just an oxidation flow reactors. Applications, of course, simulation of a chemical process is something very obvious. You can also study the evolution of particle properties, and this can have, for instance, some potential impact in climate because of the change of fortune. For instance, you can have these health studies. Also, you can use it for calibration purposes, like I showed you before. And also, you just have a new generator for particles that you can have used for very different purposes. They say, for instance, for filter testing, if you're thinking about the industry. And I am also trying to go back and we are trying to start a European project again, where maybe in 14 years from now, we'll end up having a standard that just atmospheric aging and not only have a hot filter that samples your emissions. So at the moment, we have been working more on this. Well, we, our partners mostly. And the nice thing is that, like I said, there are different groups that are using this throughout Europe. And they report also that they can simply take the set points that were introduced by this publication by Metas, and they get the same kind of particles, same results, no matter if they're in the UK, in France, or in Sweden. They can reproduce this. So this, of course, makes us happy. If you want more information, I mean, the two papers that I mentioned with the description of the instrument and description of the properties are here. In the Aerotox project, there are also some health studies, not only the one that I showed you before. And this was probably is going to be published very soon, or I hope so. And if you need more information, please contact me or contact also our partner at Metas. And here I just want to acknowledge Aerotox, which was a project that helped us finance the development of this instrument. And with this, I would like to hear your questions, and thank you very much for your attention.
Thank you for a great talk. For audience, if you have any question, please raise your hand, or you can leave your question on chat. Yeah. Maybe people need some time, so can I ask the first question? So I'm curious, like, so you say in the last slide you mentioned like you can also see the climate intervention or something, implication for climate, like radiation or something. So with that regard, can you also simulate some environment in like upper troposphere or stratosphere? Because I guess some researchers are nowadays interested in geoengineering, like releasing some aerosol in that environment, and what will happen, we don't know yet, something like that. So I'm just curious.
No, I mean, this is yeah, I mean, this is a different kind of study that you're mentioning. I mean, what I'm saying is that I mean, I usually have this slide with me, and for some reason I don't have it. If you look at these IPCC reports for climate IPCC reports, then you can see that there is a big uncertainty in what the aerosols do, if they contribute to heating or to cooling.
Oh, okay.
And
yeah.
a lot of this is due to these processes that happen in the atmosphere. And like you saw in these optical properties, you can change from a particle which is mostly absorbing light and contributed to heating to a particle that is mostly scattering light and maybe even contribute to cooling. So it's more this kind of effects that you could study and see if there is a difference depending on the kind of precursor that you're using. I mean, this is more what I meant. Yeah.
Thank you so much. Any other questions? I guess you.
Yeah, there is one question.
There's one question. Very nice talk. Thanks. The day-to-day repeatability was pretty good. What was the variation like within a single test point? How much would it vary within, say, an hour of aerosol generations?
Well, I mean, yeah, Constantina is, I mean, you can see it here. But I mean, this is, yeah, I mean, this is the size, but you can see here, it's the same day, all the points. So you can see that there is a small deviation. For instance, here, the first day, the set point was not so stable. I mean, usually it's more stable. You have the concentration that your target is. I mean, here was a little bit away. So you have this kind of, sometimes it's a kind of variation in the amount of precursor that you're dosing. And if you see, for instance, here, this is the variation in size, but this is actually very difficult because measuring particles which are between 10 and 15 nanometers with an SMPS. I mean, what's the variation which is due from the production of the aerosol, and what's the precision that you have from the SMPS? I mean, at the end, you are also trying to fit together these points in order to get the geometrical mean diameter. But I mean, still, you can see here in terms of size what the difference is. I mean, also here is maybe a little bit more. Yeah, from 110 to 120, but this is like 10 nanometers. So the difference in coating here is 10%, let's say, the most. I mean, here is maybe more easier to measure with the SMPS than here. So this is just to show you the points without the error bars, just the raw points, every single measurement. So these are several, let's say, it's probably 1 hour of measurement, maybe a little bit more. I don't know. I mean, this is stable that we probably could measure 1 hour and then change to the next set point and measure 1 hour. And yeah, I mean, at the end, you see 1, 2, 3, 4, 5, 6, 7 set points on one day. 1, 2, 3, 4, 5, 6, 7, 8 set points on one day. So you can do that with this very easily.
I also have a question in this slide. So what I've learned from the textbook is if we increase the relative humidity, then particle can grow with effluence, and so even though there is some hysteresis. But in your result, with the relative humidity increase, the particle size actually decreased after the certain point. So do you have any guess why that happened?
Yeah, for sure. Yeah, I mean, we are producing ozone with the UV lamps, and this ozone is interacting with the humidity and is creating OH. But then this equilibrium between the OH and ozone, it doesn't hold forever, you know. I mean, the lamps have some limit. If you will, we have five lamps, so we can turn on one or the 5. I mean, I think here I'm guessing the 5 lamps were on. If you reduce the amount of UV, then it will also probably this decrease will happen even further. You know, if you could increase even more the amount of ozone, then you could probably even go further. But it just is this equilibrium between the amount of OH and the amount of ozone, and this is something I in one slide. I mean, it was mentioned by the University of Basel. No, it's further down. Yeah, sorry. By the University of Basel, and we just did like this. But what they want to do is they want to change the lamps. The same lamps that we're using, which are mercury lamps, can have a coating so that you can only have the non-ozone generating lamps. So for them, it's important to try to generate SOA that is mainly produced by hydroxyl reactions and not so much by ozone reactions. So, and this is also something that you can do. It's simply you open it, change 5 lamps, and then close it, and then you are good to go. Of course, then because you are not producing the ozone, then you need to dose other substances. I mean, they also consider putting in some nitrous oxide to the second dosing part of the reactor to study the different pathways. So they want to see what's the difference if they are doing mainly ozonolysis, OH, or this, or a combination of the 3 of them. And this is something that you can do.
Yeah, this sounds good. Yeah. We have a couple more minutes, so do you have any more questions from audience? You can unmute or you can leave your question on our chat. Folks in Illinois, UIUC.
I want to add, I mean, we are not a company, we're a university.
Yep. Mm-hmm.
So we let's say per se don't sell devices, but we build several units because we want this device to be used. So we have built 7 in total, and we always keep one of each generation for us, you know, just to be able to see what's going on if somebody has a problem, also to test. But we are, until we find a commercial partner, sadly we don't have a spin-off to produce this commercially. We are open to collaborations. I mean, it's not, we are not a company selling you a product, but we can improve it, and with the help of others, and even produce one prototype for you. Mm-hmm. So if somebody's interested, please really feel free to contact me, and we can discuss how we can do. I mean, we are also happy to lend one of these units to somebody for some experiments, in order to figure out if that's something that they could use in their laboratories.
Yeah, that would be really great. Mm-hmm. Yeah. And so people can spread the word about that. Yeah.
Exactly. Yeah.
Great opportunity for interesting.
Or if a company is listening to the presentation and is interested, then of course also feel free.
So with that being said, I guess we can conclude our seminar today. So thank you for coming today, and please give a big hand to our speaker, Alejandro Keller, again. And it was really nice to see you, everyone. Thank you so much.
Thank you very much.
Mm-hmm. Thank you.
Well, thank you for the great talk. That was really wonderful, Alejandro, and great job, Mano, with the discussion. It was very exciting, and so thanks for joining us.
Mm-hmm. Thank you. Yeah, I hope I can visit like Europe for like EGU or whatever, and try to reach out to you. Yeah.
Oh yeah, that's
I should ask
feel free to drop by in Switzerland. Yeah.
That'd be wonderful.
Very wonderful.
Yeah, I mean it's an aerosol research hotspot in Europe.
Exactly.
Yeah. I mean, not only Switzerland, yeah, but also Switzerland. Yeah. Do I need to stay here to discuss something with you? No. Okay.
No, we're all set. So yeah.
Okay.
So, yep.
Thank you very much.
See you.
See you.